everyone. I hope you're doing well. Today I will talk about the action that happens in the cells and why is it important and how is it important. I will talk about the action first at the level of outside the cell and then the action that happens inside the cells. Okay, so outside the cells, we know that at organism level, we are the ones who are responsible for all the action, right? So if today I am talking to you, how am I talking to you? I am acting, like I'm talking, right? I'm doing that act of talking, or you are able to listen to me, so you are listening. That is an action that we do, right? And that is the basis of everything that happens around us. So inside cells, when we talk about the cellular environment, the whole soul biomolecules that are responsible for the action inside the cells, those are proteins. So basically, if I could simply explain to you what are proteins, they are the workers of cells. So we have a large army, and that is a very, very diverse army of cells that is basically the protein repertoire of cells, or proteome as they call it, right? And what exactly are proteins made of? They are made up of amino acids, right? Each amino acid is a different kind of a structure that gives itself a unique identity. And when these amino acids join with each other, they form a polypeptide chain. And when this polypeptide chains join one after the other to form long chain of amino acids, it is called a protein. And if you see here that there are different colors and different these amino acids are of different colors, different shapes, and so what decides that what kind of amino acid will it be? That will be based on the inherent structure of amino acid. If you see on the right hand side, we have a carbon. This is the basic amino acid structure, and there is a carboxyl group there is an amino group there is a hydrogen that is attached to the central carbon, and there is this R chain, and this is the R chain that gives the amino acid its inherent characteristic feature that will decide what will it do, who will it attract, who will it love, who will it dislike, who will it hate. So basically, the overall kind of a fate of the protein is decided by the amino acids that are there, and amino acid character is decided by the individual R group that is there, right? And if I could talk about the fate, so what will decide the amino acid fate? So which means that different amino acids will form different kind of a sequence, and the order in which these amino acids are joined together will be different, and that will decide the fate of the amino acid, right? So if you see from my previous example, these are the different amino acids that are there, and the sequence in which they are arranged, meaning will the red will come first, or the blue will come first, or the blue will come first, or the red will come first, meaning the sequence of the amino acids, the repetition. Is there only 3 repetition, or is it a long chain of same repetition? That will eventually decide the size of the protein, the structure of the protein, the function of a protein within the cell, and also the capacity of a protein, right? If we talk about the structural levels of the protein, so I am actually going to kind of stop my screen sharing so that you can see me nicely, right? And I'm going to explain this with a simple, I know you have a hands-on session this Sunday, watch out for that, but please bear with me for 2 minutes. So basically, this is, if you see this necklace that I have, every bead here represents an amino acids that are there, and of course these are all white, but imagine these each bead of this necklace is a different kind of an amino acid, meaning different color of an amino acid, right? So the sequence and the inherent nature of the amino acid will decide what this protein will be. Will it be a small to four kind of a peptide, a 4 amino acid long protein, or a long protein like this, right? And this will eventually decide the fate of the protein. Now, again, coming back, the sequence of this protein is the primary structure of the protein, right? It will suppose A, B, C, D, E. This is the primary sequence, right? That will decide. Now, if I kind of get back and give you a secondary structure, if this, imagine now this polypeptide that I have is this, right? And this inherently goes like this. So why is it going up like this? Because of attraction between this carbon, this amino acid, and this amino acid, and this amino acid, this amino acid, and this amino acid. So basically, inherently, which was a plain stretch of amino acids called protein, now it is getting its secondary structure due to attraction between amino acids. I hope you're following me. Now, this is the secondary structure of a protein. Of course, there are different kinds of secondary structures also. But I'm just giving you an example with one kind of a secondary structure of an amino acid that is called an alpha helical structure. This feels like a helix, right? So this is called an alpha helical protein structure. Now, if this is the secondary structure of an amino acid, and if some amino acid here, now some amino acid here, how will it reach there? Imagine what will it have to do to reach here? It will have to fold itself so that it can interact, it can join with this amino acid, right? So what you see was initially what was a sequence of amino acids, which was called the primary structure, now then turned into a secondary structure where amino acids that were in the vicinity nearby to each other kind of interacted with each other, and then now we see this is the tertiary structure wherein this amino acid here is interacting with this amino acid here, and it is making some structure, some globular structure, right? This is the tertiary structure of an amino acid. And now imagine I have only one phone, so I could just manage this one wire. Now there are 3-4 different such globular proteins that are there. 1, 2, suppose 2 of them, right? And both of them, one protein here interacts with another protein here, so they will form a bond, right? These two independent chains that were never joined, that could never be joined, they joined together via some amino acid to amino acid residue or residue to residue interaction, and this is the quaternary structure or the tertiary structure as we call it. Okay, so I'm going to again start.
That was wonderful, Zile. That was so animated, so involved. Absolutely. So, absolutely. Go ahead.
Yeah, so I'm just going to reiterate what I said just now. This is the primary structure of an amino acid, which is the sequence of amino acids, and there are different types of amino acids inside the cells. And what decides? The R group that is present decides what kind of amino acid, what would be its likes, what would be its dislikes, right? How will it behave? And overall, then this kind of coils on another called secondary structure, which further folds on to the tertiary third degree structure, and which further kind of folds up to a quaternary structure that finally forms a protein that we see in the cellular milieu. Okay, so if you have any questions, maybe Karishma or Snehal, we can stop, or we can, shall I carry forward?
We can take maybe some questions.
Sure.
I think one question that somebody asked was, can you cut proteins?
Yes, of course we can cut proteins. So, in cells, we have molecular scissors. So now imagine this is not your house. So in your house, you have scissors, right? But in cells, we have molecular scissors, and these are called proteases. So these are specific enzymes that cut proteins, right? And that cut protein that only after a specific amino acid. You remember the amino acid that was there? So suppose there would be amino acids called serine proteases. Serine is a type of an amino acid, so it will cut after serine amino acid. It will try to find. Suppose this is a polypeptide chain. It will scan through this polypeptide, the molecular scissors that is there, and it will, once it finds a serine amino acid, it will cut there. And that's how proteins function. If I have to degrade, suppose the work of this protein, whatever it was, in the cells it was synthesized and then it got over. Now what do? We don't want to pile up everything. You have garbage in your house, right? You throw it away. So that is, in fact, that is the cell's molecular mechanism to cut proteins and then reuse them.
So Soumya asked the question, why do amino acids attract? It's basically their chemical composition, Soumya.
Yes.
Depending on the R chain, that it determines their interactions. So what produces amino acids? The carbon, hydrogen, oxygen, nitrogen in our body keep synthesizing these amino acids. What produces proteases? Our cells make proteases, right? It's all within the cell. That's what Zile was saying. This is the action happening in the cell. So we cut proteins, we break proteins for different functions. But when you cut your nails, you're cutting a protein. You're cutting the protein keratin.
Yes.
Also, Snehal, when you fry an egg, we had something called denaturing, but you're changing that protein. Oh, Zile is back. Zile is back. Okay, go ahead, Zile. We were just holding fort. What organelle makes protein? Okay, Zile will talk about the synthesis of proteins now. Good question, Kavish. Good question.
Okay, so if we talk of proteins, where do they generally come from? So this is the central dogma of life. So you know that there is a DNA or our hereditary material, right, that we get from our parents, and that DNA has the code that decides the sequence of amino acids that will be there. Of course, that there are kind of secondary structures and secondary mechanisms like epigenesis that is there that decides what kind of protein will be there and how much will it be there. But inherently, going back to the source, the proteins come from DNA that transcribes to an mRNA, and then this mRNA is read to be made a protein, which this process of protein synthesis is called translation, right? From an mRNA, we make a protein. Okay. Coming to how do proteins look, and first we will talk about how do we look, right? So if you see Karishma, we have a lovely photo of her. This is how she looks. This is how Snehal looks. This is how we look. And each one of you would have a different kind of face and everything. So this is how we look, and this is how proteins look. This is how proteins look on our plate. Maybe from, I mean, if you see meat or you see cereals, these are all proteins, our muscles. It has a protein called myoglobin. Inside the cells, proteins are of diverse shapes. And the original idea that because there are so many permutations and combinations possible, which leads to different proteins in the cells. And again, it gives us a benefit because you can perform so many functions, and each protein has a very specific function. So if we talk about the views of protein, what does a protein look like? And I will first use an analogy here. Like, it all depends on from where you're watching the protein, right? So if for example, if you look moon towards the moon, and if you look standing on Earth, you look towards the moon, and it will look something like this to you, right? But if you go on the moon and you look, it will look completely different to you. So basically, the bottom line is that proteins come in different shapes and sizes, and they look very different also, right? But the kind of instrument that you're using, the kind of magnification, meaning how much you're zooming in or how much you're zooming out, that decides what the protein would look like. And of course, there are, if we talk at the molecular level, at the cellular level, there are different ways based on the different models or different ways we view proteins. So if you see, this is one protein, this is a quaternary protein, because there are 4 different individual polypeptide chains here also, but these are different views of the same protein. Coming to the protein-protein interaction and what is basically protein-protein interaction. So if you see this protein that is there, it has it is made up of 2 polypeptide chains, and when 2 proteins have to interact, there is an interface that is there, right? And it is at this interface that these 2 proteins interact, and these proteins interact at this interface via specific amino acid. This is red protein, this is green protein, and it leads to bond formation. And again, it depends on who loves what. If this amino acid loves this amino acid, if they are oppositely charged, one is positively charged, like lysine, which is a positively charged amino acid, the other is aspartic acid, which is a negatively charged amino acid, it will form an ionic bond with the other protein, right? Or there might be a hydrogen bond. So depending upon what is across, the bond will be formed. Acid loves, the bond will be formed. And mind you, the strength of all these kinds of bonds is very, very different, right? So yeah, and I mean, why are we talking about this? Why is protein conversation so much important, right? In the cells, if we talk, it is very important because it is a very fundamental process. Meaning, it will lead to all kind of processes. For example, DNA replication, right? Or the structure of the cell is maintained due to proteins. What it is like we have gates on our in our homes. So the gates of the cells are made of proteins called channels. So these are all proteins. The enzymes, there are so many enzymes. All enzymes in the proteins are proteins. So inside the cells, there are many diverse kinds of functions that proteins do. Outside the cells, if we see at the organism level, it leads to movement. My muscle, I'm able to move my muscle. Muscles are also proteins, right? It can move due to protein-protein interaction, because that protein-protein interaction can lead to movement via generation of some force. Okay, so maybe if you want to stop for a few questions.
Sure, Zile. So Snehal, see, Ishwar asked a question. Zile, maybe you can just go over trying to get examples from them. What are the uses of proteins in our body? Maybe we can just reiterate this. So, what are the uses of protein? For example, our hair, keratin gives our hair structure.
Keratin. Yes, the hair structure is keratin is a form of a protein. Myoglobin, or you know blood, right? That is composed of hemoglobin. That is a 4 polypeptide chain. So so it acts as a connective tissue in our cells. Basically, proteins have multiple functions, and it depends on at what level you're seeing the function. Are you seeing the function at the cellular level inside the cells? So if we talk about inside the cells, it has all the cellular processes like DNA replication, transportation, or someone asked about the scissors, right? Protein cutting, that also function is maintained by proteins. When has to be done, DNA replication, that is also maintained by proteins, because there is a sequence of events. There are different proteins that interact with each other and that finally lead to the function that needs to be done.
So, for example, Zile, hemoglobin is a protein. It carries oxygen.
Right.
Iron component carries oxygen. So, example, hair protein gives structure. Right. Hemoglobin carries oxygen. Protease is a protein that cleaves protein. It's an enzyme that cleaves protein. So guys, maybe you come up with some examples. Yes, Sridhari gives a great example. Sorry, Srihari gives a great example, Snehal. Yes, hard keratin in nail. Nail is a protein.
Yes.
Soft.
Yes.
Soft. Yeah.
Anushka
and
has
you
a
see-
very nice question. Anushka
Yeah.
asks, is there something that doesn't have protein?
Uh, Fats. I mean, so if you see an egg, I'll give you an example. An egg, there is albumin protein outside the white part, but
Yes.
inside the yellow yolk is fat.
Yeah. Right.
So it's not exclusive, but there are domains that are there. Like there would be some domains, or in our cells that we have tissues like adipose tissue, right? That is fat.
Which we
So,
all
and-
want to make into protein. That is the goal. Right, we get that. Yes, muscle is all protein, Bhargavi. Muscle is all protein. Yeah,
Myoglobin. It is a myoglobin. The protein that is there is called myoglobin.
absolutely. So what is the protein in egg, guys? Zile, maybe let's ask them this. What are the
Yes.
protein? What is the name, the scientific name of the protein in egg? Egg white. Okay, albumin.
Very
Brilliant.
good. Excellent. Excellent.
Very good, Ala. Maybe we come up with few more questions. So we said,
Okay, guys, there's one protein that all of you have definitely heard of recently. Okay, because of whatever's happening in the world right now. And your parents have gone and gotten a injection recently that helps them make these type of proteins. So what are these types of proteins? Exactly, Adrit. Antibodies, right? All these
Yes.
antibodies we've been talking about
Very smart,
that are
huh?
supposed to help us fight this virus.
Yes, yes.
They are proteins.
absolutely.
And these are special kind of proteins that help in detection of other proteins.
On the
right?
virus.
They're very specific. Like this protein will only detect this. It won't detect this. It won't detect it. Very, very specific.
So what protein does it detect on the virus, guys? Your parents must have come home talking about it. This antibody
We've talked
protein.
about this.
Okay, yes, Snehal. Snehal
We've
is very TTAS has done it. Yes, the antibody
Antigens,
proteins.
yes.
But to which antigen? There is
Which
a comment-
antigen on the virus?
Yes,
Something on the coat of the virus.
correct. Guys, you've heard this. Outer covering, protein jacket.
Yes.
Come closer. Zoom in more. Which part of the virus?
Spike protein.
Yes, Pranav.
Wow. Very good,
Athansh,
Pranav.
yes.
And Ashant also. Yeah.
Athansh, yes. Great.
Yeah.
Okay,
Spike
so Ala
protein.
had one question, which is a very nice question. I don't know if you might be coming to this later, Zile, but she asks, can proteins also be harmful to us? Can there be diseases that are caused by proteins?
Yes. There is a disease, in fact, called prion disease, wherein the protein. So basically, when I was explaining the folding of proteins, so if this folding of protein doesn't happen in a highly regulated manner and in a way it should happen, so proteins will be misfolded, right? And then they won't perform their function. In fact, they will get accumulated. So the accumulation of proteins leads to prion's disease.
Guys, have you heard? Yes, Adrit. I was just going to say that. Zile, he just said it. Is it mad cow disease? Yes.
Yes.
It is mad cow.
Yes.
And it was very much there in bovine communities in the UK, actually. And so they had these very specific rules about eating beef and things like that in the UK. Yes, it is mad cow, which is a protein-mediated disease. Great example, Zile. Thank you. So read about this. The reason it's called mad cow is because these prions deposit in the brain of the cow and they behave erratically. So let's
Okay.
name some proteins, guys.
Yes.
Aditi, let's name. Come on, type. Let's name some proteins. Come on, let's go on. We can do this for a few minutes. I'll go with the easy one. Albumin. Ala says keratin.
Keratin, lectin.
Very
Okay.
good.
Milk.
Very good. Hemoglobin. Okay, go on, guys.
Yes,
Go on.
important.
Keratin.
Peptin.
Peptin.
Okay.
Wow.
Okay. Protease.
Yes.
Myoglobin. Pepsin. Hemoglobin. Wow, look at this, guys. What else?
Lactin. Yes.
I'm not sure.
Antibiotics.
Antimicrobial peptides, maybe. Yes. But
Yes.
yeah. Tubulin. Very
Tubulin,
good. Actin.
yes.
Yes.
Galectin.
Myosin. That is the in the muscle. FTSZ, wow, extremely technical. Very good. Phe is phenylalanine. Phenylalanine is the amino acid. Okay.
Like protein, yes, that's a protein.
Trypsin, yes, is a protein. Very good. Pepsin, trypsin. So many. These are all. Lactose is a sugar. Lactose is a sugar, but the enzyme lactase is a protein.
Protein. So basically, all enzymes are proteins, but all proteins are not enzymes. Right?
Yes,
I get
absolutely.
all enzymes are proteins. So, but the vice versa is not true, because proteins have so diverse functions. They can be enzymes. They can be just transporters. They can be for anchorage. There are so many functions.
No, ATGC are not proteins. They are bases.
They are
They're like
nucleotides.
amino acids, but they are not protein. Correct.
Yes,
Okay.
yes.
Cardiotropin. Wow. Wow. Look at that.
Lactin.
Yes.
Okay.
Yes.
Okay.
Yes. Okay.
All right. So we can move on and we'll come back to some questions in some time.
Yes, yes, sure. Okay, so the next question that we will kind of address is, we talk to each other, right? We talk to each other, we communicate with each other, and we communicate with each other in different ways. So the proteins also talk to each other or communicate to each other, and we will try to address a problem that we would have, is that if we want to find out if there is this A protein that is there in the cells, there is this B protein, and there is this C protein, right? There are three proteins that are there, and I want to find out if this A protein interacts with B protein or not, or this A protein interacts with C protein or not. So there are two questions that I have in my mind. Does A protein talk to or interact with B protein? And does A protein talk to or interact with C protein? So in order to address this, I actually designed two experiments, right? In order to kind of address the question one, I designed experiment number one, and to address question two, I designed experiment number two, and I will show you parallelly how we actually address these questions. So we go to the lab and we experiment. And while there are many different ways to study or test if two proteins interact with each other, I'm going to tell you one such method, and that is enzyme-linked immunosorbent assay, or in short form, it is called ELISA. So, as I told you, that we'll run two parallel experiments. Experiment one to answer question number one, that does A protein interact with B protein? And in experiment two, we will try to find out if A protein interacts with C protein. So what will I do first will be to kind of coat one of the proteins that I need to test on this plate that is called ELISA plate. You see this black kind of a rectangle that is there, and this denotes the ELISA plate, right? And I coat this one of the proteins that is there, and here I'm using the A protein for both the experiments because that is the common protein. So I coat this protein, A protein, on the ELISA plate. Now I want to check the interaction of this protein with B and C. So what will I do? I will add to this this ELISA plate, this B protein and C protein. So in experiment one, what did I do? I added the protein B and allowed binding into the cells, binding to the A protein. And here in experiment two, I allowed binding again with the A protein by adding this the third protein, the C protein. Now imagine if let us suppose one of the proteins never interacted and one of them did interact. So if they interact, they will bind to each other via amino acids, right? They will sit on each other and interact with each other. And here, this one did not interact with each other. So, but then there are excessive extra protein that is there. So what is at this stage, what is important that we do? We wash, we remove the excess protein that is there, the unbound protein that is there. So here, because none of the circles or the B protein bound to the blue protein, everything got washed off, right? But here, only the protein that was bound, this one remained, the additional one was washed off. Now the proteins are binded, but we haven't seen this, like we cannot see this because this is, proteins are very small. So we need to have a system wherein we see these, we can calculate or see this with our eyes, right, in some way. So what we do is we use another protein called an antibody. This antibody gives color. So basically, we in both the experiments we use antibody that is very, very specific. So in experiment 1 we use an antibody. You see, this is the green antibody, and this is different from this pink antibody. Why is this different? Because this green antibody could specifically recognize the circle that was there here in this previous there, had it been bound, and the pink antibody that was there, that can only recognize the rectangle or the square that is here. And this light that you see, this is responsible for the color. This is again another protein that is attached to the antibody, and it will give color and will help in detection. So again, like the previous step, I allowed binding, right? And only when the protein that this antibody binds to, if it is there, it will interact. And that is why the previous step that we did of washing, that was very, very important and very, very crucial. So now is the result time, right? We added one protein, we added second protein, we added the specific antibody that could detect only the second protein. And now we want to see how much of the second protein is there or got bound to the first protein that was initially coated on the ELISA plate. So for that, when we and this substrate, what it does is the antibody is attached to another protein that can results in the colored product formation. So if you see in experiment 1, there was no color development, only white liquid was there, right? Which kind of proves that the result here would be, my inference would be that A protein does not interact to B protein because there was no color development, right? But if I look for the second experiment here, if the two proteins interact, the antibody would get bound here, the pink one, and that will result in the yellow color formation. And the inference that I have here is that the two proteins interact. So visually, I could actually see, yes, the proteins interact with each other. And now, this is not an all or none phenomenon, right? I mean, the strength of the interaction can be different. So this is an ELISA strip or an ELISA plate that I'm talking about, that we usually use in the lab to kind of experiment. And here, if you see, this is very, very dark yellow, right? And this is white, and this is light yellow. So there is gradation. So which means that the strength of the interaction is also different. The darker it is, the more stronger the interaction it is. So this is the more stronger, this is less, this is less, this is no interaction. So visually it is fine, but then how will we actually quantitatively measure? This is qualitative analysis. So if we want to measure, like how, like, is it interacting only this much or very strong interaction? So that we measure by taking absorbance. Absorbance means that we have and we use this instrument called spectrophotometer, right? And the absorbance is the light passes through one side of the plate, and we see how much of it is transmitted or comes out from the other side of the plate, and that's how we intensity of the color. So suppose if I was to take an absorbance, suppose this one would show one absorbance, while this one would show zero point 0.5 or half absorbance, and this one would show zero absorbance. So which means that here the protein-protein interaction is a lot more than
Stronger.
here, right?
Absolutely, absolutely.
So if you have
Okay.
any questions with regard to this, I'll be happy to answer.
Okay, we can take some questions. So any questions about understanding, are there toxic proteins, asks Ala.
Yes, so proteins are toxic. Some of them, depending upon if they are not if they're not if they're lying out there where they're not supposed to be, their function is not there, then they will cause toxicity to us. As I mentioned previously in one of the examples called Trion's disease, right? It leads to toxicity.
Right, it can lead to toxicity because proteins are deposited in different body sites.
Yes.
Yeah,
Yes.
yeah. Yeah, many of them may not remember, but when you are born, you take a heel prick blood. Ask your parents about this. Your mothers might have been totally like catastrophized that they've just given birth to a baby whose heel is being pricked. But that is to detect the presence of an amino acid, which someone said, right? Phenylalanine. So that is to detect whether you can metabolize phenylalanine. If not, you have to be put on a special diet. So if you can't break it down, phenylalanine, an amino acid, can become toxic. So ask your parents about this, okay? Many of your parents will remember, oh yeah, and we asked the doctor, why did we have to heel prick my baby? He's just like 1 hour old. Yes, most of you have done this. So ask about this and read about it if you want. So some proteins can be toxic, harmful.
Yes.
Okay,
Yes, they can be.
Zile, you can go ahead. Substitute for protein.
Very tough question. I mean, I don't think there is a substitute to proteins because we can synthesize proteins in the lab. Like we try to do, like there is translation that is happening in the cells, but we can do in vitro or in test tube translation, right? But we try to mimic what is happening in the cells in the test tubes, in the labs.
Right. Zile, good questions. Many ask is, what is this RH? Yes, RH, rhesus factor, is a protein. It is a protein on your red blood cells.
Yes, yes.
And that's
yes.
when you become B positive. B, A, O are all types of, or rather B and A are types of proteins.
Right, right.
Yes, antigen means protein. So say I am B positive. Yes, I'm always positive. I'm B positive. That means I have the B and protein and I have the RH protein. Can anyone come up with an example of their own? Which protein will you have? O is not a protein. O is absence of A and B. AB is presence of A and B proteins. Yes, you get the gist, guys. O is absence. Oh, okay. But if you're AB positive, means you have A protein, B protein, both on your red blood cells, and that RH protein. That's why you're positive. Okay. Yes. So you see, proteins are literally everywhere. We're coming up with more examples, Zile, as the session is progressing.
Yeah.
A negative means there is no RH protein, not no protein at all.
Yes.
Yes. All right. Go on.
Okay. So we talked about proteins. We talked about protein-protein interaction, and now I am kind of drawing towards what I did in my PhD. And so basically, I worked on the parasite that causes malaria, and that is Plasmodium, right? And this Plasmodium has a very it's a very smart kind of a parasite. It behaves differently. The way it looks, it changes. And that change happens depending upon what does it need, right? So where does it needs to enter, where does it need to go? So basically movement is very, very important in the life cycle of Plasmodium falciparum parasite that causes malaria. And what happens is that if the parasite has to move from point A to point B, what does it need? It needs power, or it needs energy, right? Because that is how you can, suppose if I tell you to move from this point to this point, so you will have to apply some energy, and you'll have to push your leg up, and then you'll move forward. You'll take a step forward, right? And then you take another step forward. So this is how the parasite also moves. And why is movement important for the parasite to survive and cause malaria? Because it can enter into its safe house. That is how, because the movement allows it to enter. If it was lying there and its home is here, if it can't move, how will it enter and how will it be safe and how will it multiply? So which is why movement and power is very important for us as well as for the parasite. And again, coming to the molecular basis of it, if we see from where does the power to the parasite is achieved, where does the parasite gets the power to move, it is due to a complex that is there, which is composed of proteins, right? And this protein, there are multiple proteins in there, but there are two proteins that are very, very important, like very, very important, and that are responsible for generating the force, right? I told you here that you need some power, you need some force for movement. So the force will be generated, and this force will only be generated when two proteins called myosin A and MTIP will interact with each other, and these are nothing but two proteins, and they interact in a very, very specific manner. If you see here, this is the crystal structure. This is a very, very, crystal structure means a very, very zoomed in atom to atom structure where we know what amino acid of this one protein is interacting with what amino acid of the other protein. So it is a very zoomed in structure of two proteins of Plasmodium parasite that causes malaria. And we see here that there is this small tail that is there that fits in to the whole of the second protein, that is MTIP. So MTIP is like this. Of course, it's a huge protein, but then it has a cavity. It has a hole wherein the myosin A tail fits in here, and this is what is called the molecular motor. We all have motors at home, right? We have motors in our scooters, at our home, we have water motors. So what does a water motor does? It ensures that the water from outside their house gets transported to your overhead tank. So there is movement via the force that is generated by the motor, again with the help of ATP and protein-protein interaction. So coming back to the malaria Plasmodium parasite, as I mentioned, the parasite needs to, if you see here, this is the RBC, the red blood cell that is there, and the parasite, when it infects us, first it goes to our liver, and then eventually it attacks our red blood cells. So and the process wherein it enters our red blood cell is called invasion, and this is very, very important with respect to the overall life cycle of the Plasmodium parasite, because it is here where it increases in number. It is here where it gets its food from. So basically one parasite, if you see here, it was one parasite that infected this one red blood cell, and it went on to different stages, and then it multiplied and it bursted into multiple parasites. So one parasite results in two parasites result in four parasites, 4 in 8 in 16.
So Zile, one question Bhargavi is asking.
Yes.
Just to get it clear, if myosin A tail and MTIP attach, is it a good thing or a bad thing? Should they be attached or should they not be attached?
So basically, for the protein, for the Plasmodium to move, right, they should attach.
They should
if
attach.
they don't attach, they won't move.
Okay.
So basically, if the parasite is still, myosin is here, MTIP is there. But if the parasite has to move, this tail fits it inside the hole and generates the force that pulls the parasite.
So this is an example where two proteins have to interact for the parasite to move.
Move.
Okay.
Yes.
The home
Yes.
of the parasite, Pooja, is the red blood cell. That is the home. Yeah.
Yes.
Okay. All right.
Yeah. So basically, what again coming to the question that was just asked. So basically, so if we want to develop new medicines for treating malaria, one kind of an approach can be to block this crucial protein-protein interaction. And the approach here would be that if these two proteins, these two green and red protein, myosin and MTIP, do not interact, the force won't be generated and the protein will be paralyzed, literally paralyzed.
All right.
And there won't be any malaria. So that is what I did in my PhD, and I actually found out a new protein. And this protein is called ZA1, and it is named after my name, right? And this is a new protein, and this protein is not known. This protein was never ever known. I discovered it, and because there were multiple proteins, so there were like ZA1, ZA2, ZA3, but of all those proteins, ZA1 protein or peptide, and I'm saying it to be a peptide because it was a smaller protein, like an 80 amino acid protein. Had it been an 800 amino acid, I would have called a protein. So the length of the protein decides the terminology. But so this protein, what it does, is usually it like if you see in the parasite, what was happening, the green myosin A tail was interacting with blue MTIP that was leading to the fourth generation. And what I did, I pulled this out, this tail, and I found a new binder to this, a new protein binder to this. And so even if that binder bound, it did not generate the force because it did not interact with the downstream proteins that were there.
Yes, that was the peptide you named after you. It could bind with that tail part, and there was no generation of force. Okay.
So when these two proteins, now myosin A, MTIP do not interact. MTIP is lying alone, right? Oh, my ZA1 protein, that is my new novel, my discovered protein that is there, it interacts with myosin A tail, and this parasite movement that was very fast was basically stopped. It was stalled.
Fantastic.
Yeah, this was basically my PhD work that we published also.
Okay, Zile, there's a question from Vikram. Why ZA1 and not simply ZA? Is there a Zileanum 2, Zileanum 3, Zileanum 4?
Yes. So basically, these proteins, they were fished out from a library. There were thousands of proteins that I had synthesized, lakhs of proteins. 10 to the power of 6 is the if you want to get an idea of the number of proteins that I synthesized. In fact, I did in vitro translation. In test tube, I synthesized proteins, and I saw which protein interacted with my target protein, that was myosin A tail. And there was a sequential reduction because I had 10 to the power of 6 protein, and from that I had to get one protein.
So only the original Zileanum worked. The 99999 didn't work.
Yes.
Awesome. So young minds, you have seen a scientist who in her PhD discovered a peptide that targets something very critical for malaria, and she has a peptide named after her. So who says scientists can't be celebrities? Like next time we talk of Bollywood celebrities, I'm going to be like, you may have films, but I have a peptide.
Actually, that's a very interesting question. Where was this peptide derived from? If I could just answer this.
Yes. Go ahead, please.
As I mentioned, Kashif, this was like in cells, proteins are synthesized via translation. I actually mixed amino acids, different combinations and permutations of amino acids, like some negatively charged amino acids, some positively charged amino acids, and in different ratios, right? And the idea was to get sequence of amino acids, like different sequences of amino acids, and then when I got this library of proteins, like we have a library of books, I got a library of proteins. And then I tested one-on-one interaction, and I also did high throughput screening. High throughput screening is when you test in one go all the proteins, And that's when I found my one protein out of so many of them.
So potentially, Zile, this ZEV1 can become a medicine
Yes,
to
yes. So
prevent or treat
basically,
malaria.
yes. So basically, the proteins are also again very susceptible. So we cannot directly give this. So because there are so many protein cutters that are there outside the cells, proteases as we discussed. So we need to protect it in a nano vesicle and then give it.
And then give it.
So
Okay. But
yeah,
that PhDs who work on drug
exactly.
delivery and medicine delivery can do that. Awesome,
Yes.
Zile. Congratulations. It's 6 p.m. Sorry, we need to cut it short. It's 6 p.m. because many times the kids are using their parents' devices.
No, 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 no.
So that just sticking to time kind of becomes important. But Snehal, shall we wrap it up?
Yes. Okay.
So,
All right.
guys, tonight
So,
dinner table talk about peptide ZA1.
yes, and all the protein names you learned today.
Yes. Thank you so much,
Okay.
Zile. Thank you. It was a
Yes.
wonderful session.